Welcome to our discussion on the stage-adaptive toxin neutralization platform. This innovative framework addresses Clostridium-associated wound infections using an injectable thermogelling hydrogel for targeted toxin neutralization in high-risk wounds. Clostridial wound infections. These infections, though rare, are highly aggressive and life-threatening, with mortality rates of 20 to 30%. The focus is on toxin, a potent enzyme causing rapid tissue destruction. Current treatments are reactive, emphasizing the need for proactive enzyme-targeted interventions in high-risk wounds. Toxin destabilizes cell membranes, increases vascular permeability, and triggers inflammation. It targets phospholipid-rich membranes, causing hemolysis and vascular issues. The enzyme is stable at physiological temperatures and pH, making it a prime target for neutralization. Intervention within 24-48 hours is crucial to prevent irreversible tissue damage, especially in diabetic patients. Surgical debridement is crucial but limited by anatomical constraints and patient factors, often leaving enzymatic damage unaddressed. Hyperbaric oxygen therapy aids healing but is not widely accessible. Antitoxin therapies face production and administration challenges. These limitations underscore the need for innovative, localized intervention strategies targeting enzymatic tissue damage. The injectable thermogelling hydrogel platform is engineered for sol-gel phase transition at physiological temperatures. It transitions from a liquid at room temperature to a semi-solid hydrogel at body temperature. This reversible, biocompatible process involves polymer architectures like poly-N isopropyl acrylamide derivatives. Key properties include optimized gelation temperature, mechanical stability, porosity, and biodegradation rate, supporting wound healing. The injectable thermogelling system offers significant advantages for wound care, especially in complex wound geometries. It allows for comprehensive coverage by flowing into irregular spaces before gelling. This ensures effective protection against enzyme-mediated tissue destruction through a multimodal intervention, including magnesium oxide nanoparticle release and messenger RNA-encoded protein inhibitor expression. The thermogelling system ensures comprehensive wound coverage, especially in complex diabetic foot ulcers. It provides sustained therapeutic release for 5 to 7 days, reducing dressing changes and patient discomfort. The minimally invasive application is accessible, requiring standard injection techniques. The hydrogel's controlled release and bioabsorption support wound healing, integrating with tissue and avoiding complications from non-degradable implants. Magnesium oxide nanoparticles provide broad-spectrum antibacterial activity through hydroxide ion release, oxidative damage, and membrane disruption. Their nanoscale size allows penetration into biofilms, ensuring comprehensive antimicrobial coverage. The messenger RNA-encoded enzyme inhibitor strategy offers localized toxin neutralization, minimizing systemic exposure and immunological complications, with transient expression for effective toxin control. The stage-adaptive toxin neutralization platform combines immediate magnesium oxide nanoparticle antimicrobial action with evolving messenger RNA-encoded enzyme inhibitors. This creates a time-layered defense against clostridial pathogenesis. The platform's components, including thermogelling hydrogel and tirzepatide, are coordinated for effective intervention in various clinical scenarios, enhancing wound healing through glycemic control and metabolic optimization. The hydrogel matrix actively coordinates antimicrobial and enzyme neutralization strategies. It ensures immediate magnesium oxide nanoparticle activity, sustained messenger RNA delivery, and continued nanoparticle release. The porous structure facilitates enzyme inhibitor diffusion, maintaining therapeutic concentration. The biocompatible matrix protects components from degradation, optimizing messenger RNA stability and cellular uptake while allowing tissue integration and drainage. The RAMI-5 checkpoint framework optimizes mRNA design for toxin neutralization. Checkpoint 1 focuses on target selection, validating toxin due to its conservation across clostridial species and extracellular accessibility. Its role in pathogenesis and druggability, with a known crystal structure and zinc-dependent mechanism, makes it an ideal therapeutic target. Checkpoint 2 focuses on optimizing mRNA sequences for toxin inhibitor proteins, ensuring stability, specificity, and controlled expression. Strategies include competitive inhibitors, decoy substrates, and allosteric modulators. Protein stability is enhanced through disulfide bonds and hydrophobic core packing, aiming for 70 to 120-hour functional stability. Specificity is achieved by modifying binding interfaces to target bacterial A toxin selectively. Checkpoint 4 focuses on minimizing immunogenicity in enzyme neutralization therapy. Strategies include sequence humanization to reduce immune recognition and formulation modifications to avoid immune responses. The goal is effective A-toxin neutralization with minimal host immune activation, ensuring therapeutic efficacy in repeat applications without adverse reactions. The platform's flexibility allows for development beyond a single indication product, enhancing economic viability through diverse market applications. This adaptability supports a broader range of therapeutic uses, making it a versatile solution in medical technology. The clinical application framework targets early identification of high-risk wound populations, such as diabetic patients with deep foot ulcers and immunocompromised individuals. Stage-adaptive protocols tailor interventions based on wound risk and progression, from prevention in high-risk cases to adjunctive support in established infections, ensuring timely and effective treatment. The thermogelling hydrogel system integrates with standard wound care protocols, enhancing debridement and infection control. It complements surgical debridement by addressing enzymatic damage in deep tissue planes. Systemic management, including glycemic control and antibiotic selection, remains crucial. The hydrogel provides localized protection, supporting systemic treatments and promoting healing. Patient education on infection signs and therapy compliance is essential. Comprehensive preclinical safety validation is crucial, focusing on the messenger RNA-encoded enzyme inhibitor. Biocompatibility, local tolerability, and systemic exposure assessments are essential. Regulatory classification involves device, drug, and biologic aspects, with the Food and Drug Administration likely classifying it as a combination product. Early regulatory engagement is vital for defining development pathways and addressing jurisdiction-specific requirements. The clinical development pathway emphasizes safety and effectiveness, starting with Phase 1 studies on safety and pharmacokinetics. Efficacy evaluation faces challenges due to low infection incidence, necessitating large populations or high-risk focus. Surrogate endpoints like wound bacterial reduction and healing improvement are crucial. Post-market surveillance will monitor adverse events and resistance, ensuring real-world effectiveness. Research priorities focus on optimizing formulation parameters to enhance effectiveness and minimize adverse effects. Studies should balance magnesium oxide nanoparticle concentrations and messenger RNA doses, and explore stimuli-responsive elements for adaptive therapeutic release. Platform expansion includes targeting other bacterial enzymes and adapting for various wound types, leveraging the platform's modular nature for ongoing enhancement. Preclinical validation in animal models is crucial, focusing on diabetic and infected wounds. Clinical strategies must tackle rare infection prevention, using adaptive trials and enrichment strategies. Health economic modeling will assess cost benefits, aiding pricing and reimbursement. Real-world evidence will further validate clinical effectiveness and economic outcomes. The stage-adaptive neutralization of Clostridium A toxin via an injectable thermogelling hydrogel marks a shift to preventive care in high-risk wounds. This platform integrates thermogelling technology with messenger RNA-encoded enzyme inhibitors, addressing both antimicrobial and enzymatic challenges. The RAMI 5 checkpoint framework ensures systematic optimization, enhancing outcomes for diverse wound care populations. The stage-adaptive atoxin neutralization platform leverages established Clostridial pathogenesis knowledge, delivery technologies, and messenger RNA therapeutics. This convergence offers a unique opportunity to address unmet medical needs. With proper development and validation, it could enhance outcomes for high-risk wounds, advancing targeted mechanism-based therapies in wound care and infectious disease management. This framework explores innovative approaches to clostridial wound infection prevention, acknowledging the interdisciplinary contributions needed for success. It is conceptual, requiring further research and validation. Clinical management should follow established guidelines. The author, affiliated with Alkalili BioAI Limited Liability Company, prepared this independently, highlighting potential therapeutic opportunities. The stage-adaptive A-toxin neutralization platform is associated with CAGE code 15GR4. This code is crucial for identifying and managing the platform's development and deployment within relevant regulatory and operational frameworks.